Hello, everyone. Our presentation is about understanding the 2869 INSG mutation and its role in cystic fibrosis. So, some background info on cystic fibrosis. We read many papers to get a larger understanding about this, and such as the cystic fibrosis gene, a molecular genetic perspective, the clinical phenotypes of cystic fibrosis carriers, and the restriction site generating polymerase chain reactions for the probeless detection of the hidden genetic variation application to the study of some common cystic fibrosis mutations. And after we read all these, we found out that cystic fibrosis has many symptoms, which include chronic cough and lung infections for respiratory issues, digestive problems such as greasy stools and constipation, growth challenges, which include poor weight gain, skin abnormalities, which are salty tasting skin and jaundice, and some other complications that you could face are sinus infections, delayed puberty, and stomach pain. The life expectancy for someone with cystic fibrosis is around 30 years old. And there are many treatments for cystic fibrosis, but none of them truly cure the situation itself. And you could use medications like antibiotics to fight infections. You could use all mucus thinning agents to clear your airways, and pancreatic enzymes to aid digestion, and bronchial rate dilators to open airways. And some CTFR details include the wild type genomic DNA sequence length is 12,256 bases, and the protein length is 1,480 amino acids. So, like you guys can see, it's very big. So our mutation is the insertion of a guanine nucleotide at the position 2869 of the CFTR gene. So we searched Google and the NCBI database website listed in the laboratory information section of the LB145 website to just gain some more knowledge about what this mutation even is. So the 2869 INSG mutation of the CFTR gene caused the disease because it results in a frameshift mutation, which leads to a premature stop codon and a non-functional protein. The acronym of the mutation is the 2869 INSG, which stands for the insertion of a guanine nucleotide at the position 2869 of the CFTR gene. The acronym refers to the amino acids that were changed, the DNA bases that were mutated to lead to a codon change and amino acid change was the insertion of a guanine base at position 2869, which disrupted the reading frame.
All right, moving on to literature research. We use Google Scholar and PubMed to search research papers and reviews on cystic fibrosis and the 2869 INSG mutation. So far, we have found two research papers that discuss this mutation in cystic fibrosis. The first paper is from Science Direct entitled Restriction Site Generating Polymerase Chain Reaction, also known as RGPCR, for the probeless detection of hidden genetic variation, application to the study of some common CF mutations. The paper demonstrates an ideal experiment where they take five mutations, G542X, 2869, INSG, Y913C, N1303K, and 1717-1GA, and use PCR to screen for the known mutations of cystic fibrosis. We will use the methods and the known primer base pair lengths to develop our own experiment strictly towards 2869 INSG. The second paper is a PubMed paper review entitled A Frameshift Mutation in the Second Transmembrane Domain of the CFTR Gene, Identification, Regional Distribution, and Clinical Presentation. This paper is a review of the 2869 INSG mutation about what it is and how to identify it. We found two research papers where the authors designed a PCR based on diagnostic assays and can now identify the primary DNA sequences that should be able to work in our PCR. Moving on to the 30-day optional experiment. To further connect our own biological research with the other fields of study, we plan to generate a 30-day project that tests and strengthens our own sociological understanding of genetic diseases and evolution. In a period of 30 days, we decide one of the best ways to stimulate the breathing problems affect efficiently that people with diagnosed with cystic fibrosis have was simply breathing through a straw. Breathing through a straw is a simple method to experience restricted airflow where you breathe in and out through a straw to simulate breathing difficulties like asthma. The best way we plan to do this is for every 3 days, we would rotate people to forcefully breathe in and out of a straw for exactly 1 hour right before they go to sleep. We would determine how much harder it was to breathe and investigate potential sleep difficulties afterwards. We do this in a way that is not hazardous and safe compared to the other ideas. After the 30 days, we will have a better reflection of people with cystic fibrosis after the discipline project, and we will have gained a better ability to write and speak about health and disease.
Okay, so this next slide is our primers PCR. We read the primer design section of the lab guide and reviewed the full genomic sequence of mutation 2869 INSG. We plan to design our own primers this way, and primer number one that will only bind at a certain location in the DNA sequence if the mutation is present. Here are our primers shown. The D is the forward one and the R is the reverse one. And once we know the base sequence of these primers, we can calculate its expected annealing temperature of 52 degrees Celsius for 30 seconds. Next, we have our controls. We will also create a control primer number 3 that binds well to the normal gene sequence, which is non-mutant, at the primer 1 location. Using this primer, we will amplify a segment of lambda DNA via PCR. Our main primer, primer 1, which is the wild type DNA, has a mutation due to the insertion of guanine at the 2869 nucleotide. The primer number 3 that will also serve as the control will be the modified primer, which replaces an adenine with a cytosine. A second control primer, which is the published primer, will also be designed, and this will be used to replicate a published PCR experiment versus a CFTR mutation in human genomic DNA.
So this is our works cited page for all the sources that we've used. Thank you so much for listening. I hope you guys liked our presentation.